ماشي يعني سامعاني كده؟ اعملي ستوب شير. ماشي ستارت شير بقى. شكرا. تمام كده؟ يعني انا شير سكرين عندي مش كده؟ اوكي سوري. تمام عندك كده؟ انا كده هنا الصورة عندي كويس.
الصوت ولسه في صوت مفتوح سامعين؟ لا شغال من كده الصوت واضح.
فيديو ميوزك عايزك تقفلي الموبايل.
بسم الله الرحمن الرحيم. Ladies and gentlemen, good morning. We welcome you all here in Cairo. At this opening ceremony, allow me to gladly introduce the 7th Euro-Mediterranean Conference of Life Sciences, Pharmacy and Biomedicine, BioNat-7, organized by BioNat Global Academy. alternative or complementary medicine is different from what is commonly known as scientific medicine that emerged about 2 centuries ago through the industrial advancement, primarily in the industrialized countries. Most alternative medicines are products of the indigenous people. Thanks to immigration, their knowledge has spread to other parts of our universal globe. As participants of BioNat-7, we are, through this conference, privileged to have this rare opportunity to exchange our knowledge and expertise in this unique and timely field of natural products. Let us therefore take full advantage of these days that we shall spend here. It's therefore my pleasure to introduce the chairperson of the conference, Professor Ashraf Abdessalem Khalil. Ashraf Abdessalem Khalil holds a position of professor at the Institute for Genetic Engineering and Biotechnology, City of Scientific Research, located in Burj Al Arab, Egypt. Prof. Khalil published more than 50 articles in peer-reviewed journals, and he is a member of the editorial board of some peer-reviewed journals. Khalil is a fellow of the European Organization for Molecular Biology and the American Association of Cancer Research. Prof. Khalil is the founder of the Euro-Mediterranean Association of Life Sciences, EMALS. EMALS initiated the series of Euro-Mediterranean Conference of Life Sciences, Pharma and Biomedicine, BioNats, which were held 6 times successfully, in which Prof. Khalil was the key leader. In 2020, Prof. Khalil has started a new challenge and founded another non-governmental organization called BioNat Academy, which will take care of BioNat conference series from now on. Let's welcome Dr. Ashraf Khalil.
Ladies and ladies and gentlemen, dear notable speakers, dear dedicated participants. Yes, it is the 7th International Euro-Mediterranean Conference of Life Science, Pharmacy and Biomedicine, BioNat-7. Under such global circumstances, we are very glad to see you all here in this international gathering. Indeed, I am so delighted that finally our goal comes to be true. And I'm extremely happy to give a speech to you as BioNat-7 participants. For the 7th time in a row since 2009, we are here together again to enhance our knowledge in research of life science and biomedicine, and to provide the opportunities for developing a network as well as building up regional and international collaborations. Please keep in mind that collaborative research parallel to consolidation of all scientific complexity should drive the academic communities to the right direction. As such, we are certainly aware of how successful BioNat previous efforts were in addressing issues common to professionals of life science and biomedicine across the Euro-Mediterranean area, and providing a platform for enhancing communications among specialists. For the first time, the BioNat conference held as a hybrid event, including on-site and virtual activities, which gives the opportunities for out of the borders to join and gain knowledge. It is very hard work, tiresome and challenging, but it is safe. BioNat-7 has a keynote speakers, plenary speakers, oral presenters, poster presenters, electronic posters, on-site and virtual attendance from Egypt, Sudan, Jordan, Iraq, Algeria, Romania, Germany, Italy, Ukraine, Ireland, France, South Africa, United Kingdom, and the United States of America. Despite the power of promise and modern medicine, large segments of the world's population cannot or do not obtain benefit of modern medicine. More than 80% of people in developing countries cannot afford the most basic medical procedures, drugs, or even vaccines. Among wealthier populations in those developed and developing countries, alternative or traditional medicine is popular, although proof of their safety and effectiveness is uncertain. Let us hope that BioNat-7 participants will present some answer to some of these points. Up to my knowledge, this is the sole reason in non-Mediterranean and Middle East areas to have such international meeting focusing on life science and biomedicine. BioNat-7 in brief have participants from 15 countries, 35 academic and industrial institutions and organizations, 5 keynote speakers, 8 plenary and invited speakers, 8 oral presentations, more than 25 posters, 10 scientific sessions, and 1 poster session and presentation. Before giving the floor to the first session, I would like to express my sincere thanks to my wonderful assistant team. Please stand up. Also, deep gratitude to BioNat-7 honorary speakers who have directly helped us get this kind of conference off the ground. Finally, special thanks to the sponsor for supporting the BioNat conference. Ladies and gentlemen, I wish you an enjoyable and productive evening. Thank you.
Honorable BioNat-7 participants and respected audience, we are confident that this event will provide some answers to the challenges that our guests of honor have highlighted. This international event gives its participants a rare opportunity for building a strong, viable, and sustainable network in the fields of life sciences, pharmacy, and alternative medicine. To all of us here, it's a great honor and privilege to host this international meeting in the Euro-Mediterranean zone. Last but not least, the time had come and our conference had kicked off. You are all therefore invited to enjoy your time in Cairo, enjoy our BioNat-7 event. And now, we will start by our first session, the keynote session. Chairperson, Professor Ashraf Khalil, and we have 2 keynote lectures by our eminent speakers, Dr. Ibtihal Demirdash and Dr. Sameh Saad Ali. Let us now together close our hands to welcome our first eminent speaker, Professor Dr. Ibtihal Demirdash, Professor and Head of Pharmacology and the Toxicology Department, Faculty of Pharmacy, Ain Shams University. Welcome, Dr. Ibtihal. Thank you.
طب بعتذركم بس الموبايلات عشان بس السبيكر اللي مركزين معلش محدش يرد على اي كلمه جوه القاعه. تاريخ هتوصل. الدكتورة هاني مجاش رئيس قسم الأدوية والسموم كلية الصيدلة جامعة الشمس. التايتل اوف التوك Research and Drug Decision, the landscape for drug discovery and development.
بسم الله الرحمن الرحيم. بسم الله الرحمن الرحيم. حضرتك سامعيني؟ أنا مش حاسة إن أنتوا سامعيني، صح؟ حضرتك كده سامعيني؟ مش ممكن يعني عشان بس ما طيب اوكي. Dear professors, doctors, and colleagues, first of all, it's my pleasure to be with you in this conference. Thank you for Dr. Ashraf Khalil for his invitation. My lecture entitled Translational Research and Drug Repositioning, the New Landscape for Drug Discovery and Development. Before starting my lecture, I would like to ask a couple of questions. First one, how long do you think it takes for a new drug to get approved? قد إيه بناخد كمدة زمنية عشان نوصل بدراج للماركت؟ What do you think? Actually, around 12 to 15 years. تخيلوا حضراتكم تلت عمرك البروفيشنال كارير بتاخده في انك تكتشف وتوصل دراج to the market. The second question, how much investment is required to bring a new drug to the market? What do you think? Around $1 billion. يعني تقريبا مرتب 665 employees for 15 years salary annual salary. طبعا لو حسبناه بالجنيه المصري الرقم هيبقى بمرتباتنا الرقم هيبقى اكبر بكتير. So under these circumstances we can conclude that the biopharma industry is suffering and we need alternative strategies to alleviate this suffering. In this lecture within 20 minutes hopefully I will cover the following points. Definition, what is the meaning of translation research and also what is the meaning of drug repositioning, why they are important, how can we implement, and what are the obstacles. Firstly, what is the meaning of translation? Simply, translation is the process of turning observation in the laboratory, clinic, and community into interventions that improve the health of individuals and public from diagnostic and therapeutics to medical procedures and behavioral changes. I think it is very long definition and it may be confusing for some of us. So let me tell you a story about a scientist. This will further help us to understand the meaning of translational research. This scientist, his name is Dr. Michael Dyer. Dr. Michael Dyer, he is a scientist in the field of the basic neurobiology research. He worked at St. Jude Hospital, it is Cancer Research Children's Hospital at United States. He worked for 15 years. One day, two clinicians knocked him on his door and asked him to spend some time with them at clinic. اللحظة دي دكتور مايكل اكتشف حاجة غريبة جدا. أنا ليا 15 سنة بشتغل في المستشفى دي عمري ما نزلت الكلينيك ولا عمري قابلت بيشنت. بدأ الدكتور مايكل يسأل clinicians. His interest is in the neuroblastoma genes and pathway of inducing cancer. Neuroblastoma gene is a tumor suppressor gene, and when there is mutation in this gene, this will induce certain types of cancer known as neuroblastoma, in particular in the children. He asked the clinician, what is the impact of thousands of papers published in the PubMed in this field on the way of patient treatment? المفاجأة, the clinician's his answer, there is no impact whatsoever. طبعا كان المفاجأة بالنسبة لي كل الأبحاث اللي احنا بنعملها as a basic research وبننشرها ملهاش تأثير في طريقة علاجنا للمرضى. بدأ دكتور داير يناقش الكلينيشن. Why? The most important point is there is no a good animal model that mimic the clinic reality. أنا ما عنديش animal model حقيقي بيماثل ريتينوبلاستوما كانسر. فبالتالي لما بعمل assessment للدرug in vivo in experimental animal, we assess in certain situation which is totally different from the actual or the clinic situation. Dr. Dyer started to change his opinion and started to search for a model for retinoblastoma which resembled the clinic situation. He بدأ يعمل deletion the retinoblastoma gene from the mouse genome, but unfortunately there is no incidence of cancer in the animal. Why? After research, he found that there is another gene known as B107 is upregulated or overexpressed to compensate the deficiency in retinoblastoma. When he deletes both of retinoblastoma gene and B107, 50% incidence in the of cancer occurred in the animal. So he started to use this experimental animal and assess different chemotherapeutic combinations or cocktails in comparison to the standard therapy. Within 18 months, he discovered a new cocktail or combination which is more effective in the treatment comparing to the standard therapy. تخيلوا حضراتكم احنا بنتكلم كنا عن 15 years، هو قدر في 18 شهر يحقق treatment effective مقارنة بالتريتمنت الموجود. نقدر نخرج من القصة دي إن لما حصل communication ما بين البيزك ساينتست والكلينيشن قدرنا نوصل لأهداف أوضح ونتائج أسرع. So we can say that translation research is a communication between basic and the clinic scientists to understand the clinical reality of disease progression and the therapeutic intervention that will ultimately improve the human health. To further understand how can implement the translational research, I will talk about one of the drugs that discovered using this technique. It is known as trastuzumab. In the market, it is Herceptin, if you know this drug. Trastuzumab is used for treatment of certain type of breast cancer, in particular, HER2 positive breast cancer. ازاي اكتشفوا الدراج دوت؟ خلينا ناخدها خطوه خطوه. في البدايه لاحظوا كلينيكالي ان في بعض البيشنتس ريزيستنت للكيموثيرابيوتيك تريتمنت وبيكون عندهم البريست كانسر مور اجريسيف او ميتاستاتيك. لاحظوا في المجموعه دي من البيشنت ان الحاجه اللي اوفر اكسبريس هو الهيومن ابيديرمال جروث فاكتور ريسبتور 2 احنا بنسميه HER2. They started to study the molecular design of this receptor and the mechanistic pathway, and started to design and synthesize the drug that can inhibit or block this protein. This drug was trastuzumab. Then they assess this compound in vitro and in vivo, and in clinical trials reaching the effective treatment within few years. إذا by translational research قدرنا نحقق new drug في مدة زمنية أقصر. So we can say that translational research is designing drugs to act at a specific protein or other targets directly linked to the disease, rather than evaluating thousands of compounds at thousands of targets. I hope you can understand now what is the meaning of translation research. So going to the other term, which is drug repositioning. Simply, drug repositioning is a process of identifying new therapeutic use for old existing available drugs. يعني بمعنى بسيط احنا بنكتشف علاج جديد او استخدام جديد لادويه بالفعل هي موجوده في الماركت. طيب ايه مميزات ان انا امشي في هذا الابروش؟ تخيلوا حضراتكم ادويه بالفعل موجوده في الماركت، يعني احنا عندنا الدراج بروفايل including pharmacokinetic studies انا مش محتاج اعملها تاني، safety studies انا مش محتاج اعملها تاني، فارماكودايناميك لو بيشتغل على نفس التارجت انا مش محتاج اعملها تاني، انا محتاج بس اثبت النيو ثيرابيوتيك يوز. By this way, this strategy is highly efficient, time saving, low cost and minimum risk of failure. واعتقد ان الكورونا واللي حصل فيها الدراج بوزيشننج بدا ياخد وضع كبير. صحيح هو الفتره الاخيره قبل الكورونا الاف دي اي في اخر سنتين قبل الكورونا 30% من الدراجز اللي كانت ابروف كانت دراج بوزيشننج، لكن مع الكورونا الاهتمام بالموضوع ده اصبح اكبر. As an example for drug repositioning, Oanofin, or in the market, Reduer. Oanofin is a drug used for treatment of rheumatoid arthritis. أنا متشكرة ليكي لأن الضوء كان هيتعبني. Oanofin is a drug used for treatment of rheumatoid arthritis, but now it can be used for treatment of chronic lymphocytic leukemia. Dr. Scott, who is responsible for this discovery, he said that what we did was go from in vitro experiment directly into the patients. بعد ما اكتشفوا ان الدراج ده can kill this type of cancer cell, they start clinical trial directly to assess the efficiency, and now it can be used as anti-cancer drug. Actually, we have two strategies when we use drug repositioning. The first one, we have the drug, it is known, and we have the target site, but we use the same target site for another therapeutic use. This type of drug repositioning is called on target. يعني ببساطة الدراج بتاعي على نفس التارجت سايت اللي بيشتغل عليه اي بروتين ايا ما يكون ريسبتور ايون شانل انزيم وات ايفر، انا بكتشف another new therapeutic use. As an example for this situation, minoxidil. Do you know minoxidil? Minoxidil is a direct vasodilating drug, واحد من important vasodilators. It acts through I can't remember now, it's okay. It is a vasodilator. مش بس vasodilator through the same mechanism. The mechanism of minoxidil vasodilation is through potassium channel opening. By the same mechanism, they use it for hair loss, which is Rogaine. Do you know Rogaine is aminoxidil, but as aminoxidil sulfate, which is the active form. So we use the drug by the same target site to another therapeutic use. Another approach is we have the drug and we have a target site, but we discover another target site for the drug to give another therapeutic use. In this case, it is known as off-target. As an example, aspirin. Aspirin is well known as non-steroidal anti-inflammatory drug and used for inflammation and pain. But another mechanism, it has anti-platelet aggregation effect by another target. So it can be used, it is used now for another application as anticoagulant. How can we achieve drug repositioning? As I said, we have on target, off target. We can say that in on target, we have the target. So we make screening for the effect, the other effects that can be achieved on the same target. This type of approach is known as activity-based approach or experiment screening. When I don't know the target or we need another new target, in this case, we use the in silico-based approach. In silico-based approach, it depends on identifying the molecular mechanism of the target and the mechanistic pathway to approve the therapeutic effect. Both of them are used, but which one takes longer time? Activity-based technique takes longer time because we make screening for different subjects or this. Now, what is the difference between translational research and the drug repositioning and the traditional methods that we use? Usually, we synthesize a number of compounds and screening for their activity for different uses. So we started with large number of compounds. When we go to the preclinical phase, the aim of the preclinical phase is to obtaining information about the efficacy of the new compound, toxicity, and pharmacokinetics. When we reach this information, we can achieve what we can say proof of concept. كلمة proof of concept يا دكاترة هي المرحلة ما بين البريكلينيكال وفيز 1 كلينيكال. لما بنكمل المعلومات الخاصة بالفارماكوكاينيتيك فارماكودايناميك اند توكسيسيتي بنحقق حاجة اسمها proof of concept. بالإضافة لكده preclinical studies are important for optimization and improving bioavailability. الفورمولا that I will use it in clinical trials should be tested firstly in animal experiment. After achieving the preclinical step, we go to the clinical trials. I think you know that we have 4 phases in clinical trial. In phase 1, our aim is to check the safety of the drug. أهم خطوة في الفيز 1 أنا بجرب على فولنتير نطمن إن الدراج السيف وأشوف إيه السايد إفكتس اللي ممكن تظهر قدامي. The second phase أو phase 2 is important for assessing the efficacy of the drug. In this case, we tested in the patient. In phase 3, we confirm both of effectiveness and the side effects and compare with the standard therapy if we have standard therapy in this situation. After approving the efficacy and safety, the drug in this situation can be entered to the market, which is phase 4 after approval from authorizing agencies such as FDA, EMA, or in Egypt, EDA, Egyptian Drug Agency. مهم ان انا اوضح 2 terms الناس اللي بتشتغل في drug discovery and development عارفين ان دول ملفين كبار. Number 1 investigational new drug application. بعد ما بخلص phase preclinical studies انا بجهز ملف. الملف دوت بيبدا اتقدم بيه للجهه اللي will approve الدراج. بعد ما انتهي من phase 3 another application new drug application برضه بتقدم بيه. طيب انا ليه بتكلم الكلام ده كله على التراديشنال ميثود؟ التراديشنال ميثود دي احنا اتفقنا بتاخد اراوند من 12 ل 15 سنه. يا ترى مين اكتر فيز فيهم نقدر نقول هي المعوق لينا او اللي بتاخر عمليه اكتشاف الدواء؟ تتوقعوا مين يا دكاتره؟ فيز 3 فيز 1 فيز 4 actually it is the preclinical phase. احنا بنبدا بعدد ضخم جدا من الكومباوندز بعد ما بيدخل preclinical studies وبندخله على الكلينيكال واحنا مطمنين على السيفتي والافيكاسي. نفاجئ ان 30% فيلير نتيجه التوكسيسيتي اللي بتظهر كلينيكالي and 60% فيلير نتيجه ان الافيكاسي مش جيده. عشان كده NIH اعتبر 80 to 90% project failing in preclinical phase اللي بتاخد تقريبا around 4 years بتكلفنا around 10 million اصبحت تعتبر valley of death. المنطقه اللي بيموت فيها الامل في ان احنا نحقق الدراج. How can valley of death be replaced by translational research؟ سواء translational research او drug repositioning نقدر عن طريقه نعمل بريدج يخلينا نحقق efficacy with low toxicity في الكلينيكال ستاديز. زي ما اديت لحضرتكم المثالين اللي فاتوا في حاله التراستيزوماب، في حاله دكتور داير لما اختار الكومبينيشن طلع افضل من اللي كان موجود، هو قدر يعدي بالدراج او الاستادي اللي هو عاوزها تو اتشيف كلينيكال سكسس حقيقي. طيب احنا عايزين نعرف ازاي هنقدر نحقق ده. Both of translational research and drug repositioning as approach محظوظين في الوقت الحالي because we have advanced methods for assessment. As an example, molecular docking. لو في حد من الكيمستري هنا عارف ان احنا اصبحنا دلوقتي we can predict the نقدر نقول the complementary between the ligand أو drug and the target site. من خلال Human Genome and Proteome Projects دلوقتي احنا أصبحنا عندنا علم كامل بالجينز، الترانسكريبشن اند ترانسليشن باثوي بتاعها بيوصلنا لايه، فاصبح عندنا الديزيزز، كل ديزيز نقدر نقول ايه الجينيتك اسوشيشن الموجوده مع الديزيز ده، وبالتالي نقدر نحدد التارجت بتاعنا الصح علشان تو سينثسايز دراج يقدر ينجح في الكلينيكال ترايلز. باثوي مابينج network analyzing using genetic protein or disease data can add identification of drug repurposing. Retrospective clinical analysis. What is the meaning of retrospective clinical analysis؟ زي ما عملوا في التراستوزوماب، هم لاحظوا الأول كلينيكالي إن البيشنتس اللي عندهم positive HER2 عندهم ريزيستنس للكيموثيرابيوتيك دراجز. طيب احنا بقى محتاجين نعمل سنسز لدراج كان انهبت او بلوك ذيس ريسبتور، فبالتالي قدروا ينجحوا بالتراستوزوماب. يعني انا اقدر اقول لحضراتكم ان بالنسبه لي انا حتى كطالب بتوعي في الكليه دايما كل سنتين ثلاثه بنحدد نفسنا استراتيجي جديده في شغلنا. انا متفقه معاهم السنه دي كل اللي هيسجل معايا في الماستر والبي اتش دي هنبدا الاول بريتروسبكتيف كلينيك داتا اناليسيس عشان نحدد التارجت صح، بحيث نقدر نوصل لدراجز مش مجرد اخد ماستر او بي اتش دي، احنا عايزين نوصل لابلايد ساينس، نوصل لدراج سكسسفولي ان كيس اوف كلينيكال ترايل. انا خلصت الشيء دي، انا قربت اخلص. طيب اوكي. The most important problem still until now is the availability of predictive preclinical animal models. وده اللي خلى ال NIH لما انشأ سنتر كان رقم 27 National Center for Advancing Translational Science او بيسموه ال NCATS، كان هدف السنتر ده حاجتين اساسيين. نمبر 1 انه يكسر البارير بين البيزك ساينتست والكلينيشن وان هم يبداوا يتعاونوا مع بعض، فكان بيعمل اوورد معروفه ب كلينيكال اند ترانسليشنال ساينس اوورد. حوالي 60 انستيتيوت بيتنافسوا ان هم ياخدوا هذه الاورد. الاورد دي الهدف منها ايه؟ ان احنا نمبر 1 we need development of model system for drug testing. الموديل ده يكون شبيه بالموجود في الهيومن، وبالتالي اقدر احقق نسبه نجاح في الكلينيكال ترايلز بعد كده اكبر، وكمان اقلل تعرض البيشنتس لدراجز بنوصل في الاخر ان احنا نفاجئ بيها انها توكسيك وانها مش جيده. طيب النيو ابروتش ده قدر يحققه ازاي او بيشجع ليه ازاي؟ عن طريق انه يشجع الباحثين to develop genetic engineered mouse mimic the clinical situation. The other option واللي انا متوقعه ان معظم حضراتكم ممكن ما يكونش عنده فكره عنه رغم انه في الخارج اصبح ليه اهميه كبيره، هي المايكرو تشيب. سمعنا عن الاورجا مايكرو تشيب؟ كم واحد سمع عن الاورجا مايكرو تشيب؟ 2 3. What is the meaning of organ microchip؟ اسمحوا لي in 2 minutes هنتفرج على فيديو عاملاه الانكات بتعرف بيه اللي حققته من خلال المايكروتشيب.
Several thousand diseases affect humans, of which only about 500 have any treatment. Getting a drug through the current development pipeline takes too long, is too costly, and the vast majority of new therapies, about 95%, never make it past clinical trials. Drugs that seemed promising in early studies using cell and animal models often don't have the same response in humans. We need an alternate research model that accurately represents the human response. NCATS, the National Center for Advancing Translational Sciences at the National Institutes of Health, is supporting research to make this possible. Through the Tissue Chip for Drug Screening Program, a collaboration among NIH, DARPA, FDA, and researchers nationwide, NCATS is supporting research on bioengineered devices that mimic the function of human organs. The goal is to use these tissue chips to test the safety and effectiveness of potential new drugs, weed out those that don't work, and pursue ones with the most promise. The chips are fabricated from a flexible plastic with ports and channels to allow nutrients and oxygen to pass through them. Unlike cell culture and animal models, the innovative design of tissue chips replicates the complexity of the human body environment. One example is a liver chip, which includes multiple cell types that are organized much in the same way as they are in the human liver. Features such as blood flow can be mimicked to recreate what cells experience in the body. The complexity of these chips generates human-specific data, which enables researchers to better understand how the liver responds to a drug and if it is toxic. Critical data before testing in people. By connecting chips that represent various organs into a full human body system, scientists may better understand how the whole body responds to drugs and if they're safe and effective. For example, chips that include additional factors such as hormones can help researchers investigate differences between male and female responses to drugs. The ultimate goal is to use these chips in a variety of ways, including to advance precision medicine, test for environmental toxins, and develop disease-specific models such as for cancer and rare diseases. By developing, demonstrating, and disseminating tissue chip technology, NCATS is improving health through smarter science.
زي ما شفنا دكاترة في الفيديو، مايكرو تشيب هي عبارة عن هيومن سيلز، ممكن تكون ليفر، كدني، برين، لانج، وكمان سبلاي باي بلاد سبلاي كأنها موجودة جوه الجسم. ايه أهمية المايكرو تشيب؟ الحقيقة لفترة قريبة ما كانش بيستخدم غير each organ separately. فكان دايما في انتقاد ان ده زي انفيترو، مفيش الهيمودايناميك للجسم. حاليا زي ما حضرتك شايفين بقوا يعملوا كونكشن ما بين الاورجنز كانه موجودين داخل الجسم. ايه فايده ان احنا نستخدم هيومن سيلز؟ لو انا بدور على تو اسيست دراج ميتابوليزم، هل الافضل نعمله اسسمنت ان انيمال ولا ان هيومن سيلز باي هيومن سايتوكروم بي 450؟ بالتاكيد باي هيومن. لو انا بعمل اسسمنت لتوكسيسيتي اوف سيرتن دراج، احنا عارفين ان في حاجات ممكن تبقى توكسيك لينا وما تكونش توكسيك للانيمالز او العكس، يبقى هل الاسسمنت باستخدام هيومن سيلز افضل ولا انيمالز؟ لا اقدر اقول لحضراتكم ان التوجه الموجود عالميا طبعا في الدول المتقدمه سواء اوروبا او امريكا او كندا 2050 سيختفي استخدام حيوانات التجارب في الاسسمنت اوف دراجز. هو ده التكنولوجي اللي هيمشي بيه بعد كده في المستقبل. اوكي. according للتكنيكس دي اصبحنا ممكن نعمل حاجه اسمها target product profile. What is the meaning of target product profile? It is a blueprint بيشمل كل المواصفات الخاصه بالدراج، فارماكودايناميك، فارماكوكاينيتيك، ميكانيزم. كمان بيشمل مدى احتياجنا للدراج دوت كلينيكالي، الماركت بوتنشال، التكنيكال بروبابيلتي اوف سكسس، بحيث ناخد قرار قبل ما ندخل كلينيكال ترايل جو او نو جو. فبالتالي بساعد على اني ما ادخلش وادفع تكاليف اكتر وفي الاخر اوصل بفيلير مش بنجاح. علشان الوقت اخر حاجه احب اتكلم عنها الاوبستكلز اللي بنواجهه للترانسليشنال ريسيرش اند دراج ريسيرش. الحقيقه في جهتين اساسيين احنا كباحثين وكاورجنايزيشن بننتمي اليها، وشركات الادويه او الباي تكنولوجي اللي بتنفذ الادويه. نمبر 1 بالنسبه لنا culture and mindset difference between basic science research and clinical research. احنا محتاجين نغير مفاهيمنا سواء احنا كبيزك ريسيرشر والكلينيشن. دايما لما تيجي تتكلم مع الباحثين في البيزك ريسيرش هتلاقيه دايما معني جدا انه يدور على الباث ويز وازاي يعمل لها اسسمنت وقد ايه بارامترز اقدر اقيسها. الكلينيشن ما بيدورش على كده، بيدور امتى في الاخر الدراج دوت will be effective عندي ولا لا. احنا محتاجين نقرب الفكرين عشان نقدر نحقق drugs in the market بشكل اسرع. علشان نقدر نحقق ده كمان lack of understanding about the science of translation. نسبة الناس اللي عندهم awareness بالترانسليشن ريسيرش مش كبيرة، محتاجين نوسع الاويرنس. A shortage of qualified investigators. زي ما حضرتك شفتوا اصبح عندنا تكنيكس كتير ادفانسد، احنا محتاجين ريسيرشرز يبقوا qualified في الديفرنت فيلدز دي. Organizational structures and the incentives that don't encourage the teamwork essential to translational science. طبعا مش عايزه اتكلم على لجان الترقيه والقوانين اللي احنا بنمشي بيها وسياسه الاسر الوحيد ولا اكثر من اسر، فيس اسر، بوست اسر، والكلام من ده. كل ده طبعا اجينست ان احنا نشجع الناس تكولابوريت مع بعضها. طيب الجزء الثاني اللي يخص الشركات، ايه الاوبستكلز؟ نمبر 1 بيتنت كونسيدريشن. انا لما بعمل ديسكفري a new therapeutic drugs for market drug، البيتنسي هتكون منين؟ دي واحده من المشاكل اللي بتواجهنا. نمبر 2 لو ايكونوميك ريتيرن. لما انا اكتشف نيو ثيرابيوتيك يوز، الدراج موجود في الماركت بالفعل، لما بيجي البيشنت ياخده هل لازم ياخد الدراج بتاعي انا ولا ممكن ياخد الدراج بتاع الشركه الثانيه؟ يبقى اذا الايكونوميك ريتيرن ممكن ما يرجعليش انا، ممكن يرجع لشركه ثانيه. نمبر 3 فاينانشال ريسك. الحقيقه الدراج ريبوزيشننج بدايته كان الهدف منها ان احنا نعالج الرير ديزيز او الديزيز اللي ما بتحصلش غير في عدد قليل من البيشنت، فبالتالي شركات الادويه ما بتهتمش بيه، فدورنا ازاي ندور على نيو ثيرابيوتيك يوز فور ماركت دراج. ده فاينانشال ريسك الشركه بتحسبها، انا دلوقتي هشتغل في نيو ثيرابيوتيك يوز، هل هيرجع لي الانفستمنت اللي انا اخدته ولا لا؟ فدول 3 اوبستكلز بتقف قصاد ان شركات الادويه تتشجع انها تمشي في الاتجاه. لكن اكيد علشان نحقق سكسس زي ما احنا شايفين البلوكس دي، لازم هيكون في تعاون ما بين الاكاديميك انستيتيوتس، البايوتك كومبانيز، الفارماسيوتيكال كومبانيز، وكمان النون بروفيت اورجانيزيشنز، المؤسسات الخيريه اللي بتوجه دعمها لابحاث معينه. لو قدرنا نستفيد من هذه المؤسسات في دعم الترانسليشنال ريسيرش والدراج بوزيشننج ولو في الاتجاه اللي هم عاوزينه، يبقى احنا هنساعد على ان احنا نوصل لسكسسفول دراج ديفلوبمنت. اخر حاجه I hope this lecture encourage basic biomedical research to consider the translational potential of their work. اتمنى النهارده يعني اكون احدثت شيء من التغيير في اهتمامات حضراتكم، وانكم وانتم بتفكروا في ابحاثكم للترقيه او في الاشراف على الطلبه او في اي نوع من الابحاث، البحث ده هيوصلني لايه؟ Together we are stronger. Thank you for good listening.
Thank you, Dr. Tam. Unfortunately, we don't have time for questions, so maybe we can discuss with Dr. Tam after the lecture of Dr. Sam. The next keynote lecture presented by Dr. Saad Ali. He is a of tumor biology research program in Cancer Research Hospital.
57357.
Okay. The title of his talk is role of oxidative stress in COVID-19 associated mortality.
Assalamu alaikum. Good morning, everyone. I will have to speak in English because there are a lot of non-Arabic speakers listening to us, I don't know whether in the podium, but maybe online. So I think I have to speak in English, unfortunately, but I'd like to first send a great gratitude for those martyrs that sacrificed their lives for us to live. Today is the 6th of October, and I think we all owe some remembrance for those who sacrificed their lives for this country. I remember them a lot this morning because I was also passing through the ring road, which is maybe harder than the Parleave line, because I had to spend about 2 hours in my trip to this place. So, well, I mean, I'm here, but maybe it will reflect on my nervousness and my, so I'm trying to calm down. So the story, let me just give you a brief introduction on our maybe around the story that's now under consideration for it's under review in one of the journals that I don't want to disclose because we don't know whether it's going to go through or not. But it's a new angle to the story of COVID-19. We are putting our hand on a kind of a novel aspect of the pathology of COVID-19. The story started with a question, a kind of legitimate question. For critically ill COVID-19 patients, why some of them survive and others do not? Although they are similar in sex distribution, in age distribution, in even the protocols of treatments that they are receiving and the length they have been living with the virus. So there are plenty of things that are similar between survivors and non-survivors. So we decided to design a study. I will tell you a little bit more about it. I think I will browse quickly through the introduction because maybe you don't need this. Of course, the crisis of the COVID-19 continues to hit us, and every time we think that things are relieved, another new wave starts with all of the economical and social impacts that is hitting everywhere in the world. I'm giving you an update on the profile of infection. Let me just, I'm just browsing quickly until last month, maybe middle of last month. We are approaching about 5 million casualties of this infection, this pandemic. We are approaching also something like 250 million of infected persons, confirmed cases. And as you can see, every time we think that we have reached the final wave, which defies actually epidemiology as well, because we are continuing with ever increasing size waves, and the virus keeps mutated and bringing up into the table some variants that are really giving us very hard time. So it's just like it is reminding me of the story of Sisyphus. I don't know whether you heard about this. This is a character in Greek mythology. This is the guy who was punished by gods, carrying over this huge rock, and every time he probably close to the top, it's falling down again. It's just like our situation our story with the virus. To put you in perspective, these are the major pandemics in the recorded history of human beings, and as you can see, the biggest one is the Black Death. The Black Death took lives of more than 200 million people, and this happened during I think the 15th century. Also, you have the smallpox, you have the flu, the Spanish flu, and you can see that the size of the pandemic is here is placed in terms of the number of people that lost their lives to the pandemic. The COVID-19 is now located, it was located in the beginning or the end of 2020 as the 9th ranked biggest pandemics in the history of human beings. Quickly, by last month, it is actually moving into the 7th largest one. This means that we are losing lives quite quickly, and nobody exactly understands why this is happening. So there are plenty of theories, and there are plenty of drugs on the market. The drugs or the vaccinations, of course, it is helping to reduce the spread. However, the lack of Nobel Prize this year for the people that developed RNA vaccines tells you that nobody have confidence about what these findings. The committee of the Nobel Prize decided to postpone awarding anything to the people that discovered the RNA vaccine. This tells you that we still have a lot of uncertainties regarding the vaccination. The problem is that the events or the physiological events, pathological events leading to death is covering a wide range of organs. The pathophysiology here includes the failure of many of the organs. I don't have time to go through each of these. However, there are like a sepsis-like failure that leads to organ failure and death. And the actual causes of this, there are some theories about what's happening on the cellular level. Let's just browse through it quickly. For infected lung cells, there are infiltration, of course, of T cells, and these T cells, they identify infected cells, and they try to make antibodies for the released viruses that actually ended up in the bloodstream or outside these dead cells. They die with a mechanism called pyroptosis, and this is due to the quick replication of the virus inside lung cells. However, these sites would recruit plenty of T cells and immune response that leads eventually to the clearance of the virus. This is in an ideal system or in people that are exhibiting or enjoying a strong immune system. However, too strong of an immune system or too strong of an immune response may lead to the opposite sort of fate. So, for example, here, if you have huge leakages of the viral vectors and the continuous infiltration of T cells, and the defective immune response here, in this case, may lead to accumulation of cytokines and inflammatory markers that can lead eventually to an exacerbated what's so-called cytokine storm that may lead to some organ damage and death eventually. So COVID-19 severity in patients is connected quite well with overreactive host immune response. This is one of the take-home messages. And the fast release of cytokines by the immune system in response to the viral infection may lead to the cytokine storm leading to the symptoms of sepsis, which is leading eventually to multi-organ failure and death. The other thing also is that there are, in many cases, the uncontrolled inflammation, as I said, may lead to failure in hepatic and renal systems. And these are among the primary causes of the problem. At the same time, there is also what's so-called storm of blood clots. It is now established that there are downstream events that leads to increased hypercoagulability of blood and leading to storm of blood clots. And this may account for maybe about 30% of the death that happens or associated with COVID-19 infections. So during inflammation, we started this research by a given hypothesis. I'm just giving you why did we start this research and how did we design the experiments. In fact, we noted that during inflammatory conditions, platelets are activated. And when they are activated, they recruit neutrophils, and they can start a pro-inflammatory response by releasing cytokines from their granules. We thought, or we reasoned that maybe this association between platelets and between neutrophils is a mediator of the transfer of the local cytokine storm in the lung into peripheral cytokine storm that can eventually attack multiple organs. And we also reasoned that both neutrophils and platelets, they can infiltrate into various organs. So we started by this hypothesis, and we wanted to study the platelet neutrophil crosstalk and study and follow what happens to reactive oxygen species production and also cytokine level production, in hoping to explain what we see in COVID-19 patients. And this is the hypothesis put in a kind of pretty way, done by one of my students, but just for the sake of time, we wanted to look into the oxidative stress, because oxidative stress might be a direct product of this association between platelets and neutrophils, and also it may inflict oxidative stress and damage downstream. And we wanted to look into the effect. What are, of course, in the reductionistic approach in science, we want to reduce into specific questions and specific markers to go after. And from my experience and my previous work, human serum albumin is one of the most important reflectors of oxidative stress. So what we did is we wanted to see if human serum albumin is affected in these patients or not, and whether it can play a role in pathogenesis, and also it can be used as a biomarker for prediction of mortality in COVID-19 patients. This is the basic hypothesis. And also the underlying causes that human serum albumin may be playing a very important role can be understood from these facts. First of all, human serum albumin is synthesized in the liver, and it is exclusively synthesized in the liver. It is soluble in water. It is distributed everywhere, ubiquitously in the body, and it also infiltrates into various organs. More than 60% of proteins in circulating bloods are human serum albumin. It is the most important carrier of drugs. More than 220 drugs known to be associated with human serum albumin. The human serum albumin function affects their bioavailability and their pharmacokinetics and pharmacodynamics. So there are plenty of other functions of human serum albumin, like, for example, it is the most important, it's the frontline antioxidant. This is usually omitted by many researchers, but actually, because it's constituting 60% of the proteins in the blood, so it is quite abundant, even though it has only one sulfhydryl group that can be acting as an antioxidant. But this is the frontline antioxidant. The other thing is also it is important metal ion transporter, not only drugs, but also it is a metal ion transporter, and also it is fatty acid transporter. So it can affect metabolism and can affect oxidative stress, can affect enzyme synthesis. It's also very important for oncotic pressure. It's keeping the balance between the microvessels and the bloodstream, and also the flow of drugs and other factors from blood and into blood. It also, as I said, drug transporter. It also transport hormones, fatty acid transporter. And also recently there have been plenty of reports implicating human serum albumin in the coagulation, blood coagulation function, because it binds with thrombin in an inactivated way. So the hypothesis was that maybe we can, because it's central in oxidative stress, and also maybe because of the carrying of iron and other metals, it may be also affecting hemoglobin and hypercoagulability. And this is how we studied this. We recruited about 39 ICU hospitalized patients and 15 control samples of donors. Out of these, of course, when we studied the human serum albumin, we didn't know who was going to live and who was going to die. But I mean, eventually, after finishing up the study, about 16 of those were discharged, and about 23 of them died. We decided to study after collecting blood samples, we decided to study the human serum albumin, not only level, but also the biophysics, the structure of human serum albumin, trying to identify whether there have been any damage that happened to the protein. And then we studied the function of human serum albumin as a drug carrier to see, and a drug releaser to see if it really impacted this. impacted the function. Okay, so I'm not going to dwell much about this, but this is the start of the study. Is this upper right corner system? It's called electroparamagnetic resonance spectrometer. This is eventually a system that we use to monitor reactive oxygen species, but also it can give us ideas on the structural changes through some modified experiments of the human serum albumin. I will explain a little bit on this, but before going into the biophysics of human serum albumin, we did some study to identify what are the sources of oxidative stress in the blood. If you browse through literature, you will see that so many people are proposing that oxidative stress is important in COVID-19 pathology. However, nobody up to date identified what are the sources of this. So what we did is we took these blood samples, and we as you can see, the upper row is this for control, and the middle one is for survivors. It is severe recovered and severe diseased or the non-survivors. You can see that we identified this, to make a long story short, we identified neutrophils as the major source of reactive oxygen species in COVID-19 pathology. Without delving into much details, just you have to trust me about this. We did a number of studies, and as you can see, this heat map indicates the level of various subtypes of blood cells, like, for example, lymphocytes, neutrophils, platelets, and also the ROS level in all of these in neutrophils, and also a ratio that's now currently used as a prognostic and diagnostic factor for predicting severity in COVID-19. This is the NLR ratio, neutrophil to lymphocyte ratio. You can see that the red means higher values in the heat map, and this means that neutrophils increased tremendously in the non-survivors. As you can see, it is increasing somehow also in the survivors, but it is much more higher in case of non-survivor, and platelets, actually, they are reduced in our hands. DCF is almost similar to what's happening to the neutrophil population. The DCF means ROS level. It's a dye that actually stands for reactive oxygen species. And when we made a relationship between neutrophil population in these patients and between DCF positive neutrophils, we saw a very, very good correlation. And this correlation means that probably most of reactive oxygen species are resulting from neutrophils. We then tried to correlate the neutrophil count with severity, and we can see that there are correlation with severity, but not pretty much with mortality. Then we moved to see if reactive oxygen species are correlated with mortality, and there is a strong trend with mortality. However, it was not statistically significant. The p-value is 0.07. We then moved to confirm this. We devised a technique to monitor reactive oxygen species, hydrogen peroxide specifically, in plasma of those patients. And without going into much details, if you look to the lower panel, you would see that there is highly statistically significant correlation between mortality and severity, and between the level of hydrogen peroxide in plasma of patients relative to controls. And this is highly statistically significant with a relatively large number of samples. We also did some sort of imaging studies on isolated neutrophils. And you can see, if you compare the upper one, the blue staining is the nuclei, the green staining is for reactive oxygen species, and you can see that for non-survivors, there is a huge increase in reactive oxygen species relative to survivors and relative to control, which confirms the results of the hydrogen peroxide assay. Let's now see if human serum albumin predicts mortality. We monitored the level of human serum albumin, and we saw that it predicts severity rather than mortality. So we moved to study the biophysics of human serum albumin. I have to browse quickly. Just to give you an idea on what does this mean. If you look into the lower part here of the protein, it's depicting the structure, the X-ray structure of the protein, while bound with fatty acids. It has about 7 different sites for binding with fatty acids. What we did is we actually labeled or used a labeled fatty acids here. These labeled fatty acids find their ways into the sites of binding with fatty acids in human serum albumin, and we monitored the free radicals here, which is very sensitive to the environment. It can reflect structural modifications of the human serum albumin. By analyzing these spectra, we obtain multiple parameters, like, for example, what's so-called order parameter. If you look actually with eye, it's quite hard to see, but there are very important parameters that we obtain from these biophysical analyses. And when we analyze these on the large number of samples, we found that it's very hard to read, but all of these parameters indicate that there are some drops in the order parameter of the protein. There are some increase in the fluidity of the protein, and this means that there are some damages, and these damages can predict mortality and severity as well. Okay, so then we move to see if there are some changes to water accessibility of human serum albumin. And as you can see from this is an experiment that's based on loading fatty acids into the protein and then reacting this with ascorbic acid. Ascorbic acid is an antioxidant. It reacts with the free radical, this free radical, and it quenches the signal gradually. We studied the kinetics of the signal quenching to indicate how much it is easy for ascorbic acid to access, and this because ascorbic acid is a water soluble. Water accessibility can be measured through the accessibility of the ascorbic acid. And we found that there are some impairment in the water accessibility of patients, the blue and red points here. And this indicates there are some defects in the structures that is preventing water accessibility in this type. What about the drug loading and unloading? What we did is that we studied drug loading and unloading through different peaks in the EPR spectrum. One of them is coming from rigid environment, one of them is coming from fluid environment, and we wanted to ask if this can be reflecting drug association and ease of association and inclusion of drugs. And as you can see, also the blue and red one indicates that there is a significant difference between the drug loading and between also the drug unloading. In the next slide, for drug unloading, we studied this by reacting these or studying these biophysics and the contribution from the two signals at various concentrations of ethanol. And analysis of this indicates that there are some much easier unloading of drugs from the human serum albumin. So there is weaker loading and also easier unloading of drugs. Okay. The next question was that whether reactive oxygen species coming from neutrophils are the ones that are responsible for the damage observed in human serum albumin. This is an important question. What we did is we did correlative studies for the damage parameters versus the hydrogen peroxide and versus the level of oxidative stress. And as you can see, all of them was successfully indicating a strong correlation from medium to strong correlation. Of course, correlation doesn't necessarily mean causality, but we further studied this to look into the mortality now. So now we have also established a relationship that so far neutrophils are very important sources of oxidative stress. They increase tremendously in COVID-19, and that they produce a lot of reactive oxygen species that inflicts damage, sensible damage on human serum albumin. The question now is whether these damages can predict mortality. Okay? So we did mortality studies. As you can see, first we looked at what we did is that for all patients that we studied, the 39 patients, we determined the albumin level, and then we took the mean value and we stratified these into two groups, one above the mean and one below the mean. Above the mean tends to yield more survival, and below the mean tends to produce more mortality. And as you can see, the differences here are statistically significant. We did the same thing, monitoring one of the biophysical parameters reflecting the fluidity of the human serum albumin. And as you can see also, it is also statistically significantly correlated with mortality, which means that it can predict mortality. Then we moved into hydrogen peroxide, and you can see that also from hydrogen peroxide level, there are some statistically significant reduction of mortality for patients with higher levels of hydrogen peroxide in their plasma. And finally, we devised a measure where we multiplied the hydrogen peroxide in the biophysical parameter, and this led to an accuracy of more than 95% reduction of mortality in those cohort of patients. This indicates that maybe we can make a blood test that can predict the mortality of these patients within about 2 weeks. So this indicates that maybe something can be done about this. So we are suggesting in this study to explore what's so-called serum albumin replacement therapy for those critically ill people that are at risk of death, in addition to using antioxidants, specifically glutathione, which is FDA approved. I think I should browse quickly through this thing by thanking the group that actually did the work. I'm just only using their, I'm actually exploiting all of them and using their work to show it to you, but all of them are wonderful group, and they had to work immediately because our work is physiological, and they have to work immediately with fresh samples. They cannot put samples in the fridge and work later. This happens with some work, but in many cases they stayed until about 12 o'clock in the evening working on samples, and I usually sneak out and go home about maybe 7 or 8 o'clock while feeling sense of guilt. But I should thank you so much for your attention, and thank Dr. Ashraf for the invitation and for the kind invitation every year. Thank you so much.
Thank you, Dr. Sam. I have a question regarding the sampling competition. So is it random or is it design?
Well, it is. There is. This question was asked to us by the reviewing. By the reviewers of the paper. انت اللي كنت ريفيور؟ مش تقول كده طيب عشان احنا هناكل ضغط. طيب يعني the question the nature of this is usually is a kind of مش عايز اقول ان هو canned question، الاسئله المعلبه، يعني دايما لما ببعت بيبر الناس تقول how did you calculate the power of the study؟ I think the calculation of power of is misunderstood in many cases and with many reviewers. The problem is that there are plenty of problems to calculate the power or to calculate the number of samples necessary to obtain statistically significant effect. And usually this is because we are testing something that's novel. If I know what I'm going to see, I would have not gone to this study. For example, we didn't actually expect that we will see some damage in human serum albumin. Let aside how to quantify it, and then we didn't know exactly how many samples to study or to analyze to get statistically significant. However, what we did is I blinded the operators. I'm the one who's communicating with the physicians that are collecting blood samples, and we'll continue to collect blood samples until we saw a strong trend, and then statistical significance later on. But the operators themselves, they do not know. They are blinded. They don't know. Actually, all of us were blinded at the time of analysis because we didn't know whether this patient is going to die or is going to survive. You know, all analysis they are done on fresh blood samples, and we didn't know. So the analysis has been done after the continuation, after the completion of the.
Is there any questions? Yeah, Dr. Asha.
Any another biomarkers
For these patients?
Same question has been asked also by reviewers. Well, we actually tried, we have all these data, and the paper contains all the clinical parameters, including ferritin, including CRPs, including IL-6, and all of these parameters. But we fail to see correlation between these parameters and between the mortality, first of all. And this is also, if you look in the literature, nobody actually was successful in correlating mortality, for example, with IL-6 level or CRP level. The huge increase is characteristic for inflammatory response in general, but it is highly variable between patients. It depends on the regimen of treatment, and it has been quite variable in our case also because, we don't know. This was determined at a given time point. Maybe it is different from the time point when we collected blood samples, so variability can happen. And we couldn't see any consistent correlation between IL-6 or CRP or any other inflammatory marker and between mortality.
Or
lymphocytes and neutrophil count can predict severity, but it cannot predict mortality. So what we also analyzed the lymphocyte level, we analyzed platelet level, we analyzed most of the subsets, and they were successfully predicting the severity, but not mortality. So lymphocytes, the drop of lymphocytes starts to be seen quite well in moderately ill people, and then for severe people, they are the same whether with survivors or non-survivors.
I think also it is very important about plasma therapy.
Well, plasma therapy.
Plasma therapy contains albumin.
Yes, yes, of course, plasma contains a lot of albumin. People use plasma therapy in the beginning, from infected people after recovery. They wanted to see if the antibodies can recognize the virus, and they use this, but unfortunately, it didn't go that far. In our experience, we think that the serum albumin replacement therapy, which is used in the most ill sepsis patients and other critically ill patients, it could be okay, but it's not for everyone as well. I mean, I cannot give it, for example, to moderately ill people or people that will probably find their way to survive. So it's only maybe suggested to critically ill people that have these markers that they are likely to die.
شكرا دكتور سامح. So the next session, we are 10 minutes ahead of time. So next speaker is Dr. Rashid Mehmet.